Good morning and good evening and good afternoon, everyone. My name is Seirin from Kyoto University, and I'm very happy to start our MMP kick-off seminar. Okay, so before I'm going to start the seminar, I would like to give some guidance for Q&A session. If you have a question, anytime you can add your questions in the Zoom in the menu button. So this you may see this slide and following this guide, and then you just to give any questions. Then after the talk, I will just pick up your question and I call your name. Then you can just turn off your camera or on the microphone, and then you can directly ask your question to the speakers. Okay, so the next, before starting this seminar, I would like to introduce our nice advisor. So, CJ, are you here?
Yes, I am here, Saren.
Yeah, thank you, CJ. So could you please briefly introduce yourself?
Yes, hello, everyone. It's wonderful to see many of you in the room for our first in-person event. I am CJ Musante. I'm the former Vice President of Scientific Research and Head of Quantitative Systems Pharmacology and Pharmacometrics at Pfizer, currently retired, but still serving in this wonderful role. Thank you very much, Saren. I'm very happy to see all of you and looking forward to the seminar by Professor Tanaka.
Thank you. And then next advisor, I just attended in the on-site. So Dr. Kawai.
Thank you very much. So actually, I'm very excited to be here in Kyoto in person to see, a lot of colleagues. So I'm also very happy that, I hear that the more than 200 people, participants. So thank you very much for joining us. Thank you very much.
Thank you. Then next advisor is Professor Kawamoto.
So my name is Hiroshi Kawamoto. I'm a director of Institute for Life and Medical Sciences in Kyoto University. And I have been studying immunology and hematology, and rather in a basic research field. And I'm not good at mathematics at all, but so I'm going to make any effort to contribute to this meeting. So this, so これなんていうのかな、プロジェクトかな。Yes. So thank you very much.
Okay, so okay, it's Motoko Yanagida from Kyoto University, and I'm a professor of the Department of Nephrology, and I'm now collaborating with Seirin-san, and I'm learning how mathematics can solve the problem in the nephrology field. And precision medicine is also a very huge, big trend in the field of nephrology, so I'm really looking forward to today's talk. Thank you very much.
Thank you very much, advisors. Actually, we have two more advisors, but one advisor is unfortunately cannot attend here because of some clinic work, and the other advisors is coming. So I will introduce it maybe after this talk. Okay, actually in this seminar, we have the more than 220 participants from the 17 countries. And then, so, and moreover, the industry is 40% and academia is the 60%. So we are really happy to continue this seminar as a really global seminar. Okay, so then I would like to go to the talk of Professor Tanaka. So before I'm go to the talk, it is my really great pleasure, and I'm very happy to introduce the Professor Tanaka. Professor Tanaka is the professor of the Computational Systems Biology and Medicine, Imperial College London. So Professor Tanaka obtained her PhD from Department of Mathematical Engineering and Information Physics, the University of Tokyo. And after she got the PhD, she was appointed as an assistant professor in Kyoto University, Japan. But she had a really strong interest in the new area of system biology. So she received a fellowship from Japan Society for the Promotion of Science and joined Professor John Doyle's group at the California Institute of Technology as a visiting associate. So after that, she worked as a research scientist in the biological control systems laboratory in Reiko, and since 2009, she has held her current position at Imperial College London. She is absolutely a leading researcher in the system biology and the pioneer of the mathematical dermatology, as well as many allergic the disease. So welcome, Professor Tanaka, and we are very looking forward to your talk.
Right, thank you very much. Is this on? Thank you. Thank you very much, Seirin-san and all the advisors, and thank you for the opportunity to invite me here. I'm very honored to give a talk and talk about my work and my group's work on basically mathematical dermatology, although well, I should probably share the screen first. So the title said allergic disease, and I was thinking to talk about both atopic dermatitis and asthma, but when I began preparing the slides, it looks like I didn't have enough time to talk about two diseases. So today I will talk only about my work on atopic dermatitis. So I'm based in the Department of Bioengineering at Imperial College London. It's been 16 years that I've been leading the group. So as Seirin just introduced me, I'm not a biologist, or I'm not dermatologist. So my background is in control engineering. So the department that I did PhD was in mathematical engineering, but it's a very broad area. And my main theme was in control engineering. So the control engineering is kind of the engineering theory that can help how we design the system or engineering systems. And these are the examples. Anything that you can think of, like cars, airplanes, robots, drones, everything that you design, the human design, the engineering system, so that the systems work as you wish. So it's humans who make the design, because we make the system so that it works as we want. So the idea is to have basically the model of the physical model, and then design and decide what kind of inputs we need to give to the system so that the system works. And the idea that we had is that how about applying this control engineering theory or the ideas to understand how the humans or the biological systems work. So that's what I started doing when I moved to California. It was 2002, by the way. So I started looking at the metabolic networks, and basically the idea was to understand how the human control systems work, because it's quite intriguing that human behavior or human biology has lots of very sophisticated regulatory mechanisms. And usually we don't even think about how the systems is designed until something goes wrong, like human disease. So basically human disease is an outcome of some kind of the failures of the regulatory systems. So that's the idea. So when we think of the biological control, well, it's quite similar. Well, it can be considered as similar as control engineering. So we have that model, or we want to understand the mechanisms. The only difference is that in control engineering, we humans design the system. So we know everything from the small detail because it's humans designs. To understand the human's biology or the human body, how the regulatory systems work, we don't know how it was designed or how it was built, but we can use the control engineering ideas to understand how it could have been designed. So I put the control engineering or the models at the center, and it's a very similar ideas. You have different examples of the organs or the even human behaviors. And I think the way we can look at is to have the model and understand how the regulatory mechanisms work. And then the title of the talk today is about the personalized medicine. And when I typed personalized medicine, there are lots of images on Google that appeared. This is one of them. It's very, very static. Once you prescribe this, the combination of the drugs or the combination of like leaves, then maybe you can use it as a personal medicine. But I'd like to say that the personal medicine might look like this, like control engineering, biological control. It could be model-centered, could be something others. But from my perspective, this could be a way to go for the personalized medicine. And the point is that we really need to understand the system level dynamics. So there are two keywords here. System level, it's not about only the molecular or genetic level, but system level behavior as a human body with multiple scales. And then dynamics, it's not about the static state that we are looking at, but how the systems changes over time. So that's the point of the control engineering and the regulatory mechanisms. And we can probably use mathematical modeling. That will be the keyword of this talk today. And recently we use more and more machine learning and AI. So these are the combination of mathematical modeling, machine learning, AI, and people might say, use a buzzword of digital twins instead of saying mathematical modeling. So I'd like to focus on a specific disease, the atopic dermatitis. It is also called eczema. Well, many of you might have seen these kids with atopic dermatitis with red skin and very itchy and scratching, and yeah, it's a common chronic disease. And I'd like to say that it is actually more than a skin disease. So because of the constant itching and scratching, the patients, in this case, the babies or the children and the family have a strong, are strongly affected. And sometimes it's very difficult to even sleep, and it can lead to sleep loss and the fatigue and et cetera, et cetera. And the point is that it is a chronic disease, and this inflammation can relapse. It can be stay and non-inflammatory disease, and all of a sudden the inflammation happens, and then it can go up and down. And the problem is that currently there are lots of burdens to the patients and the families because they need to always self-manage their symptoms. So they go to the clinic and they receive some treatment, but basically they have to care by themselves. It's a huge burden for if you don't have a very clinical understanding of what's going on. So that's the basic of the atopic dermatitis. And I had some personal interest in atopic dermatitis, and that's where I started working on this specific disease and tried to understand how the disease happens, how we can treat, or how we can control the symptoms, and eventually how we can go towards a personalized medicine. So the challenge is to understand the disease pathogenesis here is that, so basically it's a system level disease. So it's only on the skin, it appears on the skin, but it may be like barrier and immune regulation is also involved. And we need to understand system level dynamical changes over time. And when I say over time, it could be like hours, days, weeks, or even years. And there are lots of data which follow the symptoms over like 20 years and how they change. And it is also known that there are lots of patient variability. Some people can benefit from some treatments and some are not. And also the causes of the disease may be different depending on the patient as well. And that also means that the pathogenesis is very complex, could be affected by some environmental factors, and it is affected by the barrier and immune reactions, et cetera, et cetera. So the whole point is to understand disease pathogenesis is very challenging. And how we can use computational approach to tackle this difficult task to understand the complex pathogenesis of the atopic dermatitis. And I follow these four basically steps to achieve that. The observation, modeling, prediction, and control. So firstly, observation, we need to understand what's going on. So it was like what was happening or what is happening. And then it also means that you need to collect lots of data to understand what is happening. And then the modeling, making the model, so integrate all the information you could collect and integrate to make a model. It could be mind map, it could be mathematical model, but some kind of the computational framework that will allow you to understand or allow you to extract the information from the data that you collected. So that corresponds to the why. Why this is happening, why what is we are seeing is actually happening. And then once you make the model, you understand what's going on, and hopefully that will help you to predict what will happen in future. So learning from past, what is happening, what was happening, and understand why, and that will allow us to predict what will be happening in the future. And if we can predict what will happen in the future, that also means that we can control the situation, because we can predict, using the model, we can predict what will happen, which means that we can design the inputs that can prevent the worst case scenario to happen. So that corresponds to the treatment design or personalized treatment design if the model is more personalized. So there are these 4 steps: hindsight and get the insights, and then give the foresight. So that's the basic idea of the computational approach. And then when we talk about the computational models, so it's always the same. We want to collect lots of data. Can be anything, like from experiments, in vitro experiments, in vivo experiments, clinical trials, any kind of things. And we want to integrate all the data together. And when we say computational models, basically there are 2 big categories. One is called mechanistic models. In other words, sometimes some people say it's white model, and another one is statistical model, black box model. So the mechanistic models can reveal the causality. So collecting all the information and want to understand what causes what. And here you can see the block diagram or the network structure, which integrates or which suggests what affects what. If this gene is expressed, then this protein is produced, etc. So this causality. Another one is statistical models. It's more like a data-driven, and it's also called black box model. So you get the data, and then basically what it's looking at is to identify the association between the data. It can be statistics, the traditional statistics, or AI or machine learning corresponds to this category. So it's not to understand the mechanisms, but it's more to find the association hidden in the data that you collect. And it's not like one is better than the other, and we should definitely use the both. So we can do the model-based machine learning. That is probably what we should aim for. We want to understand the mechanisms, we want to understand the causality, but we can also use lots of statistical models and association of the data so that we can effectively integrate the data that you collect. But my background in control engineering usually use the mechanistic models traditionally. So that's what I will start talking about first. And when you want to make the mechanistic models, as the figure shows, you first collect lots of pieces of puzzle. And depending on the modeler, there are lots of different ways to integrate and making the models. It's like, yeah, either you make the human form or cube, or sometimes even with the same kind of pieces of puzzle, you might want to make the sphere, and depending on who makes the model and what you want to gain or what you want to understand from making the model. And in my group, I really want to understand what makes a difference between health and disease. So in terms of the regulation. So this is one keyword, and that guides what kind of models we want to make. And then I'm also very interested in understanding how these different dynamical behaviors appear from the regulatory mechanisms. So like homeostasis and oscillation, what causes to these different dynamical behaviors. And the third one is a strong keyword for my group personally, that when you look at the biological control mechanisms, often the case we can find the positive and negative feedbacks, and they are quite well balanced. That's a stark difference from the control engineering systems, because engineering control systems, because it's very dangerous to use positive feedback. Because when you do the positive feedback, then the small error might be accelerated and emphasized, and that can cause the system to become unstable. But for the biological system, it uses positive feedback so that the small noise can be amplified. So it can help to use a small amount of the data or small amount of the noise, but by combining with a negative feedback, these noise will not be leading to the instability of the system. So it's a quite important characteristic in biological control, and these positive and negative feedback can be always found in lots of systems, and that can also be a key to find different dynamical behaviors. So on the right, you can see an example of the system that we are talking about, just to give you a flavor, what kind of things that I'm talking about when I say model. So it can consist of different layers from the skin barrier, SG, SB, and then also about the cells, inflammation cells. And the processes that are happening at the different scales can also vary over multi-time scales, from seconds, minutes, hours to days. Then, so this is the first model that we developed back in 2017, and it was we developed this model to understand how the eczema starts and progresses to allergic types. So basic idea is that we wanted to have a very minimal processes that can explain what's happening in atopic dermatitis. So here we basically think of the triangle. The main first one is environmental factors, like a pathogen or any environmental factors that cause the inflammation. And then there's a skin barrier, which interacts as between the environment and inside the body. And then the immune responses. And these three components are interacted with each other. And on the left-hand side, you can see that when the pathogens come into through the barrier, it causes the inflammation by activating innate immune receptors, and it causes immune responses. But when the immune responses become activated, then it can try to eradicate the pathogen. So that corresponds to negative feedback on the left-hand side in green. on the right-hand side in pink, you can see that there's a positive feedback. That's a kind of intriguing. When the inflammation happens, well, by activating the innate immune receptors, actually barrier becomes damaged. And when the barrier becomes damaged, there are more pathogen infiltrating to the barrier. And that causes the pathogen load inside the barrier to become larger. So that corresponds to the positive feedback. So basically, the inflammation level is determined by the balance between this positive and negative feedback. So that's first. That's the first characteristic of this system. And then interestingly, on the left-hand side, immune responses, and we often use MMP, antimicrobial proteins, and then we know that some eczema patients have some genetic variation in MMP expression. And on the right-hand side, it is about the barrier integrity. And we also know that some eczema patients have a filaggrin mutation, which corresponds to the barrier integrity. So this positive and negative feedback corresponds to the two main genetic factors that are known for eczema. And another interesting point here is that basically the innate immune receptors or inflammation corresponds to the switch. So when the pathogen load or the stimulus becomes high and high, at some point, the inflammation happens. So the upward black arrow. And then once the inflammation happens, even if the pathogen load becomes slightly lower, then it stays, the inflammation stays on until it goes, the pathogen load becomes much, much lower. And then finally, the inflammation stops. So this goes up and then to the left and then going down. And in mathematical terms, it shows the bistability because there's a region of bistability in the middle that there are off state and on state, both are stable and can be observed. And similarly for the Th2 response, that the x-axis is that the number of the activated dendritic cells. So when the dendritic cell numbers increase, then the Th2 responses becomes on. And once it becomes on, it's very difficult to stop it. It's almost like irreversible switch, not quite, but it can be like an irreversible switch. And then it also shows the bistability. So we consider there are two switches. So I changed the figure in the middle. So there are first switch about inflammation, and then second switch about the Th2 response. And the point is that these two switches are actually connected. The first switch corresponds to the yellow inflammation. And while the inflammation is on, shown by yellow, then the dendritic cell numbers increase. And then when the yellow one becomes off, then the dendritic cell numbers become off. But the dendritic cell numbers on the second switch, the Th2 response switch, has some threshold value. So when the dendritic cell numbers go above certain threshold shown by the dotted line, then the Th2 response becomes on and then stays there. So basically, we can say that the disease phenotypes can be described by the on-off states of two switches. So two switches on and off. So 2 by 2, and there are 4 disease phenotypes that we can talk about. So that was to understand how the disease happens, and then it becomes Th2 responses happen as well. But it cannot answer why there are long-term behavior of the eczema. So we decided to look at more detail of what's actually happening at the barrier, and then we found that there should be another switch. And therefore, we now call it a triple switch mechanism for long-term behavior of eczema. So basically, if the first switch is inflammation, as before, second switch is about Th2 response, but the third switch is about the skin barrier and the keratinocyte differentiation. Because when the barrier, the barrier doesn't happen immediately, but it has to start from the basal layer. And the basal layer, the keratinocyte differentiate and then go up and up to become the healthy barrier. And that also have some switch behavior. When the skin barrier becomes very low, then the switch happens and the differentiation starts. And when the skin barrier is very healthy, that differentiation rate becomes lower because it doesn't need much differentiation to happen. So this is the third switch that corresponds to the long-term behavior, because this differentiation takes time when it happens and go up to the top layer of the skin. And then what we can see is that if this third switch is malfunctioning, which means that the barrier is damaged, but differentiation doesn't happen properly, so these people will have difficulty in regulating the long-term behavior of composing the healthy skin barrier. So this is now in preparation. That's why I'm a bit struggling, because this is the first time that I'm talking about this, this result. Anyway, so now that we have 3 switches, and when there were only 2 switches on and off, and on and off. So there are 4 states. Now we have 3 switches, on and off, on and off, on and off. So there are 2 by 2 by 2. So there are 8 states of the switches. So basically, the white shows the off state, and black shows the on state. And there are 8 different transition states. And we want to see how people, or how the model moves along these different states. Obviously, when all the 3 switches are on, it means that the disease is much severe. And when all the switches off, it means that it's very healthy. So what we did is to take a few main parameters and then look at how the system behavior changes. So we did the bifurcation analysis of the model, and then identify what happens when one switch becomes on and other switch becomes off. And then by looking at the bifurcation analysis, we identified there are basically 9,264 possible transitions. So there's 1 shown here, and then 2 more shown here. But we could identify exhaustively what are the all possible transitions that can happen. But then when we have more than 9,000 transitions, what can we do? It's too much. So what we did is to binarize the switching transitions. So either quiescent state, non-active state, and active state. So we say that when more than 2 switches are on, it means that the state is, well, the inflammation is active, or the eczema is active. And then when there's only 1 switch, it's often the case that we can convert back to non-on state, so 000 state. So in that case, we assume that it is inactive. And by doing this, each of these more than 9,000 possible transitions can be described in this kind of disease activity. So when like non-active and it suddenly becomes active, or active and non-active, and back and forth. So these are the disease activity trajectory. Now the y-axis is at 0 or 1. 0 corresponds to non-active, and 1 corresponds to active. Still, we have more than 9,000 states trajectories, and that are too much. So we did the K-Medois clustering to cluster into 4 subtypes. So either no AD, so now the x-axis is time, so there's no AD at all, or permanent or persistent AD, like go up and stay, or intermittent, go up and down, or transient, it's just go up and then stay low. And by doing this, we can actually create virtual patients. Now that we know only there are 4 subtypes, so we created the virtual patients by choosing the model parameters and create the behavior which corresponds to each of these 4 subtypes, 4 times 80K, so 320K virtual patients. And then calculate the how many % of the patients had the symptoms and compared with this published cohort analysis. So there are lots of cohort analysis which follow the patients over about 20 years, and I see how the percentage of the patients changes over time. And this is a result. So the solid line with thick or the darker color corresponds to the model behavior. And the fainted colors, no, sorry, dotted lines, not the dotted lines, the thicker lines, thicker colors corresponds to the model behavior, and the fainted color corresponds to the cohort-based analysis from these 5 published papers. And remarkably, actually, it was quite surprising for us as well, but the model behavior matches quite well, and also the studies from 5 different cohort analysis matches very well. And here you can see that the time x-axis is in years. So that gave us some kind of reassurance that our triple switch model can capture the long-time behavior of atopic dermatitis of which was captured in cohort analysis. So what we did is that now that we know that there are 4 subtypes, and for each subtype, we can choose the model parameters and then see how the parameters differ between these 4 different subtypes. So for the non-AD, permanent, intermittent, and transient, and x-axis corresponds to different parameters, and y-axis corresponds to different parameter values that we can observe for each different subtypes. And we run 11 million virtual patients. And that gives us an idea of what are the main features that makes a difference between, for example, permanent AD compared to the non-AD, and transient compared to non-AD, and intermittent compared to non-AD. And it was quite nice to have a specific parameters that appear very dominant, which means that these parameters are responsible for this specific subtype. And for the permanent one, it was more about at the top layer, the pathogen exposure, barrier degradation, infiltration related parameters. And for the transient, in addition to all the parameters that appear in permanent AD, there's a specific feature about AMP response. And for the intermittent one, there's another few parameters that appear corresponds to the calcium sensing and the barrier formation, etc. So what we observe here is that when someone starts to having the onset of the AD, that's probably because of these a few parameters that corresponds to the barrier. And then in addition to that, if the remission, some people can have remission if the AMP response is very strong, and these people will go to the transient, go to the green one. But if the green parameter is not very strong, then people will stay in the red one and have the permanent or persistent one. But once you're on the green road, if there are these yellow parameters that becomes very strong, then it can relapse again. So then these people will appear as the intermittent subtype. So it was quite nice to have this very simple model with 3 switches, triple switches, which allow us to identify 4 disease subtypes, the model inferred subtypes, and then we can also identify which parameters are responsible for this specific subtype behavior. And from this, each parameter has some biological meaning, so we can find the geo terms that correspond to each parameter, and then from here, we can identify the AD-associated genes, and if someone is very happy to look at, then they can look at these specific genes to see if these really corresponds to the different subtype behaviors. Right, so I'd like to summarize the first part. What we did is to collect lots of data, putting them together, integrate together, and making the mechanistic model. And once we have the mechanistic model, we can create the virtual patients by specifying the model parameters and run the simulation or do the mathematical analysis. And then by doing the mathematical analysis, we can identify model inferred subtypes, and we can identify 4 subtypes. And for each of the subtypes, we run again lots of simulations and I see which specific model parameters corresponds to or responsible for each subtype to appear. And that can tell us what are the underlying mechanisms, and also potential genetic variations can be identified. And we developed this framework for the atopic dermatitis, but obviously this framework can be applied to any other disease if you can make the mechanistic model. Right, so I'd like to change the gear. It was about the mechanistic model, but when you have this kind of the clinical trials data, what can we do? Well, actually, we can't have this type of clinical trial data. This was obtained by doing the meta-analysis of clinical trial data. Why? Because each line of different colors is coming from different clinical trial. They are run for different time frame. They measure different severity score. So we need to do some calibration. And that was needed. So once you have the clinical trial data from different trials, we need to normalize so that actually we can compare all of these data and use it for the model. So that was the first step. But after this, we wanted to have the model that can explain this clinical trial data. And we had this by the double switch model that we published in 2017. So we wanted to use this to try to explain what we observe in clinical trials. And because these clinical trials were about different biologics, so the biologics are affecting on the immune responses side, so we specifically make it more detail of the green part of the immune responses. So compared to the previous double switch model, that green part becomes much more complicated. That's what you see on the left bottom of the figure, that there are lots of lines overall. And what we did is that once we developed this model, we did the parameter fitting. And then what you see on the right bottom figure is that the blue circles corresponds to the clinical trial data. These are the average over the participants. So it's not personalized, but we can have this model. And then the orange line corresponds to the model behavior. And you can see that it matches quite well, which means that there are lots of clinical trials from 9 different clinical trials, but we have one single model that can explain all the results from these clinical trials. And once you have this model, then you can do lots of simulations. So we can firstly identify who does not benefit from a specific biologic. In this case, it was dupilumab. So we identify dupilumab pro-responders by looking for which parameters correspond to this behavior, and then specifically take these dupilumab pro-responders as virtual patients, and then instead of applying the drugs that are already used, we inhibit several specific interleukin, pretending this is a new drug. And what we see from here is that inhibiting IL-13 and then 22 can achieve a very good result in terms of the EASI-75, which means that 30% of the patients who are applying, who receive this IL-13 and 22 inhibition, they achieve EASI-75. So yeah, this could be a good target. It was just the simulation results, but someone has to check it. And so another part about the design of the optimal treatment regimens. So for example, if we have this double switch model, what we can do is that we could do the prediction, but then we can identify what kind of control inputs will help disease to be maintained. So here we can use model predictive control. So what it does is that you have the model, so which means that you can try and see if we put this control input, like the control horizon, if we put this treatment, what would happen? So we can do all the predictions and for different treatment regimens. And then we can identify what would be the optimal treatment how do we call the treatment strategies? And then we can take that as we can design it optimal control or treatment regimens. So this is an example. If we want to use corticosteroid and corticosteroid, is it corticosteroid? Yes, C is corticosteroid. Then what would be the best way to achieve the maintenance of non-inflammation by doing this? So there's a theoretical framework to design the optimal treatment once you have the model. But another type of data that we obtain, and this is probably what's more closer to the actual patient's daily behavior. So this data is coming from another clinical trial, and the y-axis on the top plots, you can see BADR. It's a BADR score. The patients recorded the BADR score between 0 and 10, how bothering your eczema is today. And you can see that it's quite fluctuating. And this is a reality. Can we use the double switch model or mechanistic model to deal with this change of the BADR score? So that was our motivation. And we wanted to do the forecast of the severity scores, like a weather forecast. This is the weather forecast for Los Angeles. It's always sunny. And if it is always sunny, you probably don't even need to look at the weather forecast because it doesn't give much information and you probably don't need it. This is weather forecast from London. It varies and you really wonder whether you should bring the sweater or umbrella every day. And weather forecast will help enormously. And we know that it doesn't provide the complete information. It only gives a partial information and it may be incorrect, often the case. But still, it's very useful to give some ideas what we should do. And that's the idea of the weather forecast or prediction. But it should be personalized. And because we look at the weather forecast in London, if you are in London, but if you live in Los Angeles, the weather forecast in London or Kyoto doesn't matter at all. So it's kind of the personalized prediction that is required. And we wonder if we can do the similar things about the eczema severity. So these are the actual data from patient A and B. One is quite flat, and probably they don't need too much weather forecast or the severity forecast. Patient B goes up and down. Even it's a partial information, and even if it's incorrect, maybe it's still useful if you know that the weather forecast gives some ideas about, well, if we know what would happen, then you might want to change the activities and also the treatment style. So that's the motivation. So we had this some mechanistic model, but compared to the level of the data, the granularity of the data, we only had the severity score. It was too complicated. The model was too complicated. So we modified and make it much simpler. This is what you see on the middle. And this is called a mechanistic Bayesian model. And we use a specific algorithm called HMC to fit this data to the model. And you can see the black lines correspond to the actual data and blue shades correspond to the model prediction. So this was the first attempt, and we make it a bit more complicated because severity BADR score is a single severity score, but in reality, people measure or the clinicians measure different disease signs. So for each disease signs, we can develop a model, and that is called Eczema-Pred. It is a computational framework to predict patient-dependent dynamical evolution of eczema severity. That's because we have the patient-dependent severity, and then we want to use it. And specifically, we use the Bayesian state space models. So that's the technical details. So the results looks like this. Here again, you see the black line corresponds to the actual data, and blue shades corresponds to the model prediction. And for the moment, we achieved 70% accuracy for 4-day ahead prediction. It depends if you think this is a good result or bad result. I think we still have much space for improvement. But the whole point is that we didn't want to use just the black box deep neural networks, because we want to understand what's actually happening and what's actually, well, what the model is looking at when they want to make the prediction. And finally, we also developed this CNN network called the EczemaNet. So what we understood from here is that what's missing for the moment is the data for the eczema severity, like a daily change of the severity. So we developed an EczemaNet. It's a convolutional neural network and a computer vision pipeline. The idea is that when the users upload their photo, then this neural network can spit out the severity, eczema severity for different disease signs. And that will be very helpful because it's much easier to track their severity. And then if we combine with eczema pred, then we can predict what would happen in terms of severity. And another angle that we are looking at for the moment is about the microbiome, skin microbiome, because we know that the eczema patients often has a S. aureus infection, and maybe we can target S. aureus and use it as a therapy. So we developed this model on the top left. It's a bit more complicated. So if you remember the environmental factors, immune response, skin barrier, this triangle, in this case, this environmental factors part becomes a bit more complicated. But basic idea is the same. We identify what are the main processes. We collected the data. We make the mathematical model, and we created lots of virtual patients by changing the model parameters and do the simulation, and then identify what are the processes that corresponds to or responsible for the behaviors that we observe. And we say that not only just killing the aureus, but the dual action treatment is quite important. So targeted killing, but also the bacterial growth should be inhibited. So both should happen, and that will be more efficient rather than just killing the aureus. This also needs to be validated. The final few slides. This is the review paper that we published this year because we are looking at the skin microbiome a bit more and compared what's going on in the research community in terms of the in silico models. We found only 6 in silico modeling papers on skin microbiome compared to 55 for gut. So there's a stark difference between these two. And when we think about the computational modeling approach, there are basically 4 phases. So firstly, we need to integrate dynamical data, build the in silico models, make the prediction, the dynamical changes, and then we need to do validation. We make the predictions, but the validation needs to happen. And then depending on the results of validation, it goes back to model development again. So these cycles needs to happen. But what's the current situation is that this validation is not very commonly done. Even for the gut, about half shows the validation. Well, in one single paper, they do the validation. But for the skin, out of 6, there are only 1 parameter. There is only 1 parameter, 1 paper which did the validation part. And another big problem is that the to develop the in silico models, we need to identify the parameters, and the parameter estimation requires lots of data. For the guts, there are lots of people which use the data and share the data between the groups, and they can use it to parameterize. For the skin, most of the people have to do the guesstimate, which means it's not based on the data, but we need to guess some parameters and start from there. And that also means that the scarcity of the data is the big problem. So the current limitations, not enough data for in silico modeling approach. It's not only for the microbiome related ones, but in general, this is a key point. And especially for the microbiome, we looked at lots of data usually of snapshot. So people measure like the concentration of something, but not following time course. And time course data is really needed because we want to develop the mechanistic model that can tell us about the dynamic system level dynamics. So that's the key point. And another one is the data is coming from different conditions and strains, and that's obviously very difficult to solve. But one possibility is if you have the in silico modelers in the group, then we can already define what kind of experimental conditions would be suitable to develop the models. And the model, it's not to develop the models is a purpose, but making the model will identify and restrict what is the most informative experiments to be done. So this cycle is really needed. And the only way is to talk between the clinicians, experimentalists, and in silico modelers. So that's it. I showed some of our work about the computational approach, observation modeling, prediction, and control. For the observation, there are lots of now AI tools that allows us to do efficient data collection. And for the modeling, I showed some mechanistic models as examples, and it's a knowledge base for different data that we integrate. And then once we have the models, then we can predict what would happen in the future, and that can be done by simulation or something like eczema pred for eczema severity. And the control part, we can use simulation or eczema treat I didn't talk about, but there's another framework that we are making to develop the treatment design. Right. So I'd like to thank all my funders and my collaborators and my fabulous group. And thank you very much for your attention.
Thank you very much. Very fascinating and interesting, fantastic talk. So we're going to now is the questions time, but I just would like to first start with the advisor CJ. CJ?
Yes, hi. Thank you, Sharon, and thank you so much, Professor Tanaka. That was a fabulous presentation, and coming from a background of mathematical control theory, I love this slide. I might actually borrow it in the future because I think it really goes to where we're headed in this field. So one thing that struck me as you were walking through your presentation was, this is a very complex system with many positive and negative feedbacks and intrinsic and extrinsic factors that are affecting the outcomes, whatever type of intervention or disease progression that we're looking at. And the results of your clinical trial simulations are actually quite impressive and really not trivial. So I'm wondering if you can discuss how you go about understanding and quantifying the relative contributions of different disease mechanisms. And where you were headed to with that last part of your talk with the microbiome, that just really brings the level of complexity to an entirely different level. So I'm just wondering if you could walk us through that.
Which one? Which model are you asking for?
The mechanistic model from the beginning, where you were really kind of putting the mechanisms together and trying to understand what the relative contributions are for different mechanisms that lead to either disease progression or disease reversal if it's pharmacotherapy.
Sorry, I tried to share again. So this one.
Yes. Yeah. The early part of your talk, really, when you're starting to build a mechanistic model, how you understand the relative contributions, because of course in drug development, when we're presented with a new target, we're trying to understand what's the role of that target in the disease and does it have the horsepower to really reverse the course of the disease. So understanding the relative contribution of each of the mechanisms is really important.
That's a quite difficult question. So
yes.
Contribution for the specific drug effects or in disease pathogenesis in general?
Just in general, yeah. Just to walk through the process for people who aren't, who don't maybe understand, who haven't built this type of model before, just how difficult it is. Yeah.
Right. So I can just go through the left figure. Is that right? Yeah. So yeah, it's true. Thank you very much for your question. This is an extremely, extremely complex model. I've been looking at this over 10 years. I know everything, but it's not a very easy one to capture in 5 seconds. And sorry about that. So what we model is that there's skin barrier on the top here, and then viable epidermis, and the blood vessel and an immune response here. So 3 compartments. And at the skin barrier, normally the skin barrier is very healthy and tightly connected. And an environmental factor, the external pathogens, for example, cannot penetrate when the barrier is very healthy. But if barrier is damaged, then it can go through, penetrates into the viable epidermis where there's a receptor. And then when the receptor is received this pathogen, it's activated and leads to the inflammation. Right? So the receptor is activated and inflammation happens. But at the same time, when the receptor is activated, innate immune responses like MMP is activated and then try to eradicate the pathogen. That corresponds to the green part. And also when the receptor is activated, then there's 2 arrows. One is the proteases is produced more or activated, and then also the lipids process will be inhibited. So that corresponds to the barrier damage. So receptors activated, inflammation happen, MP tries to eradicate pathogens, but the barrier is damaged. And then when the barrier is damaged, because that Pacman corresponds to the protease that cuts off the barrier connection. So the barrier becomes weaker and more pathogens come through. When the inflammation happens, then dendritic cells are activated and move to the lymph node and cause the Th2, the polarized Th2 cells become more dominant. And then when the Th2 cells are polarized, it comes back to the site of the inflammation, so goes back here. And then actually, this also inhibits the barrier components to be produced. So that corresponds to the inhibition. And epidermis, these components are inhibited, and that also causes the barrier damage. So these are the main processes, and these complex processes are captured in this triangle. So the pathogens are activated, go through the barrier, and then the immune receptors are activated and going through inflammation. But when the receptors are activated, barrier integrity is, barrier becomes damaged, and that causes more pathogen to going through the filtration. And then at the same time, the immune responses are activated and then try to eradicate pathogens. Does it help a bit?
Yes, thank you. And I think I also see a question in the chat that's kind of getting at the question that I was asking as well. So maybe we'll get to it later, but thank you very much for your explanation.
No problem.
Thank you, CJ. And then we are going to the question in the on-site. So Professor Kawamoto, okay.
So firstly, a comment. So it is good to know that by this simple model, a pathogen barrier and innate immunity and acquired immunity, then you can make an intermittent model. Actually, so in clinical field, so we all autoimmune or allergy also very frequently go intermittent. I mean worsening and remission and repeat this worsening and remission. So it is good to predict such such movement so in your model. But I have some question. So actually, 15 years ago, when I was in Riken Institute, so one team was actually they are studying allergy and especially focusing on atopic dermatitis mouse model, and they try to make this kind of mathematical model of disease. But one point was that they have 2 or 3 models. One was JAK1 gain of function model, and another one is maybe STAT3 loss of function model. And so I do not remember which was which, but when you cross it to LAG1 or LAG2 knockout mouse, that is important for gene rearrangement of antigen receptor. So the LAG1 knockout means they cannot produce IgE, but still, so one model developed a similar dermatitis, and another model did not develop it. And so I have been thinking IgE is very important factor, but the similar symptom can be developed in IgE positive or IgE negative. And I think maybe in a clinical area, IgE dependent atopic dermatitis and IgE negative. So IgE independent one should exist. So, but your model says acquired immune or T helper or something, but did not talk about dependence on IgE. So I wonder whether IgE should be involved in this model or not.
Yes, so we thought about including IgE, and eventually we put everything into GATA3 and disease as well. So yeah, we didn't eventually include it because it was part of this adaptive immune reactions. But if we have the specific data about IgE and want to reproduce these results, then we will probably add that specific part more complicated and add that as well.
And so another point was about destruction of barrier, so damage of barrier. So actually, it is caused by scratching. So and scratching depends on itchiness, and I think it is difficult to predict or include exactly how itchy. So this status is. But I think this is a very important factor. To make the model. So how do you think about such itchiness or scratching? So
yeah, so that's another thing that we thought of doing, but we didn't because then we have to probably include the brain, skin, which is probably a bit farther away. And we thought it would be better to focus on skin first. But itchiness, if we could reproduce something about itchiness, that would be great. Yeah.
Okay, then my last point is, so 15 years ago, so that team tried to make at that time machine learning or AI was not so well developed, but tried to make computer learn many papers to make this kind of to draw this kind of model. But so the team faced the issue that some papers are correct, but some papers are incorrect, I mean false. So my question is, maybe this is a naive question, how you can find how you can learn which paper is correct or incorrect to establish this kind of model.
Yeah, that's the big issue actually. When we read the papers, we can't actually, distinguish the good and bad. So we have to talk to the experts to get some advice.
Okay.
Yeah, but this is a critical problem for us.
Even big paper.
Yes.
Like support.
Yeah.
Even Nature and Science sometimes tell a lie. So
Exactly. Yeah.
Okay.
Yeah. So hopefully this collaboration between the modelers and expertise in dermatology and the biologists and the immunologists can give some more insights about what to look at and how to combine them as well.
Yeah, thank you. And we are going to online participant, Nessie Tanya. Are you here?
Hi, can you hear me?
Yep.
Thank you so much for this really beautiful and fantastic talk. I really enjoy it. So I think my question follows on similar to what CJ was saying earlier, but specifically in your simulation of dupilumab non-responders, I think if I remember correctly, even in the case when you inhibit all of the cytokines, it seems like the EC75 responder still maxed out at about 40%. So I guess my question is, what were the important drivers of disease in the case of inhibition of all cytokines? And perhaps can you share some insights from your model on the effects of modulating additional mechanism beyond cytokine targeting to suggest what might be an efficacious new target or combination?
That's a very good question, which I don't have answers directly. So we looked at inhibiting all cytokines and then it still achieved a quite high EASI-75. Yes. Sorry, I don't remember what was the mechanisms that we tried to interpret and we couldn't identify. Sorry.
That's okay. That means I have to read the paper. Thank you so much.
All right. Okay, next we are going to just Professor Yanagita first, and then next Professor Hide.
Okay. So thank you very much, Professor Tanaka, for your great and stimulating talk. Although I'm a very outsider of dermatology and machine learning, I still enjoy a lot about your talk and learn from your talk. And I have a very naive question, outsider question about the modeling, especially about the triple switch model. And I'm interesting about the kind of the quantitative aspect of modeling and how you weigh the power between, for example, inflammation and pathogen, and how you prioritize three switches, and how you decide the duration of each step. So this is quite outside the question, but I'm interested because that I'm thinking about making some mathematical modeling with Seirin-san, and it is always a problem. And how to decide that the duration of each steps, and how you weigh that each step, and how you prioritize each step.
So we don't decide each time step because, right. So we write the equations as a differential equations. So when we solve it, already it includes, well, the solution has a function of time. So it's not us who decide the time step. How should I,
Yeah, I think it's the mathematical model actually captures the overview of all pathogenesis. So when we solve the equation, we just give one input information. Then the mathematical model actually is just like the patient's body. And there, when we get some stimulus, our body is automatically changed. So mathematical equation also automatically changed depending on the time. And then the output just shows to us. Then we analyze that. Yeah.
Say that this is correct.
So that's the validation point. So it's very difficult to validate because so the problems that I didn't have. Yeah. So for example, this is not exactly, but this is kind of the simulation result that we obtain. So we already have the time, and then once we run the simulation, it shows some behaviors like this. But unless we can collect the data which can measure all these points, it's impossible to validate 100%. So that's why we did this kind of analysis to compare with the data that is available. And if it is not inconsistent, I can't say that it is consistent, but if it is not inconsistent, then it gives us an idea and a reassurance that the model is okay so far, until we find some data that will contradict with our results.
Thank you. And we are going to the next Professor Hide. Actually, Professor Hide is today is the special advisor, and he is the really great dermatologist.
Thank you very much. And I'm a dermatologist. And another question, I'd like to add a comment, especially on the question to Tanya. And thank you very much for a very insightful lecture. And I understand that the mathematical model may give suggestions, especially about the balance among cytokines. And regarding the dupilumab refractory case, I'd like to introduce a case of my patients. He was actually refractory to dupilumab, but he got some benefit, but not sufficient. So I'd like to say that that is dupilumab failure. But I add on a cyclosporin, which inhibit non-specifically T cell functions. So I don't think that distinguish any cytokines, generally suppressed cytokines, and worked very well. Then indicating that the patients require more potent inhibition of cytokine network. So as far as I understand that any medication which specifically suppress cytokines do not work 100%, do not suppress cytokines 100%. So we have to think about the amount of suppression as well as the balance. In that particular patients, in addition to dupilumab, dupilumab cannot control well by itself, for itself, but together with cyclosporin, the patients get a very good control. That's my comment. That's okay.
Yeah, so what I was trying to say. Yeah, that makes sense. Yeah, so we did the parameter fitting, and after the parameter fitting, then each arrow has a weight, and probably that corresponds to the question from Yanagita sensei as well, that parameters can identify which processes are more important compared to the others. But we used one single set of the parameters, which corresponds to the mean behavior of the clinical trials data. But obviously, each patient does not specifically follow this average behavior, and then the parameter variance might lead to different responses. So all of these orange ones corresponds to the average behavior. But when each patient data comes in, then we might be able to identify the parameter sets that might be different from what we see from here. And that will correspond to explain different behavior that we observe for each patient.
Okay, thank you. And we are going to another question from online participant. Siranjivi Gurumani. Do you have a questions?
Yeah, hi. Thank you for the wonderful presentation. I have a few questions from the basic and from my basic understandings and stuff. So one was a follow-up question to, I mean, both of them are follow-up questions. So one is on the time scales. That is, we have different reactions that take place at different time scales. So when we are coming to the broader approximation of pathogenesis, and then you have immune response and all those things, each of those, if you speak in terms of molecular wise, they are at different time scales. Reactor ligand reactor receptor interaction is on a separate time scale, and then you have a transport of different molecules across different regions, which are in a different time scale, and barrier integrity by itself is in a different time scale. So how exactly are all of these actually captured? I understand that through parameters you will have a huge variation in values, but speaking in terms of huge difference in these timescales would actually result in values that are way small and would be negligible, and some of them would be predominantly larger. So when you're modeling across a huge timescale of this complete timescale regions, how exactly is the model actually capturing these variations across timescales?
Yeah, thank you very much for your question. So if we already know that there's a big timescale difference, then we use a quasi-steady state approximation and saying that the fast timescale behaviors already achieve the steady state when we look at the long-term, the slower timescale. So that's one way. And then after that, we do the parameter fitting, and this difference of the timescales might appear in different parameters as well. Does it answer?
So yes, so a follow-up question on this. So in from my experience with immune response models and stuff, we actually see that some of them are I mean, they are they have a stable cycle, and some of them would have, like, in terms of response toward specific reaction, you will have some are transient, that is short response, and then some of them are brief, and then it goes beyond for a longer time scale, chronic responses. So in such cases, you would actually have three variations of an quasi-state, right? So how exactly would those be reflected in the model? So in my case, it's actually a response. In immune response, I've gone through enough kappa B modeling, which is actually an inflammation response. And we would actually see a brief response, which is something about 30 minutes scale, and then a longer response, which is more than 24 hours and stuff. So
yes.
From this standpoint.
Yep.
Yes, I just want to understand, like, how exactly, since we have more than one quasi-state, how would it be reflected in the model?
Yeah, so for the triple switch and double switch models, we basically look at only the steady state. We did the bifurcation analysis, and we didn't make any virtual patients' behaviors on that. Well, the virtual patients, but look at the steady state. For the QSP model and the microbiome, well, the QSP model specifically, it is in the one single time scale, so this is not a problem for this model. But for the triple switch models, we look at the steady state. Once we do the quasi-steady state and look at the specific time scale.
Okay, thank you. We are going to the one more on-site question from Professor Kubo.
So thank you, the exciting talk. And my question is, so the our skin is in our skin, inflammation damages the barrier, and it is a basic design. And also in the gut, the inflammation makes the barrier leaky, and in the gut, it is very reasonable because when barrier becomes leaky, then maybe diarrhea is induced, then pathogen is flushed out, then the number of pathogens becomes decreased. But in the skin, I always wondered why our skin is designed as the barrier. It's damaged by the inflammation. So if in your model, the barrier is not damaged by the inflammation. Then in healthy people, what can such a model can answer such question is my question.
Okay, so right, if we assume that the inflammation doesn't cause the skin barrier damage, what would happen?
Yeah. Yes. What is the demerit of such design?
I have to check. Yeah, that's a very good point, actually. Yeah, because we can stop removing these processes and see what would happen. Yeah, interesting. Thank you very much.
Thank you.
Okay, so I would like to go to the final closing comment. The another organizer, Dr. Miyoshi, could you give a nice closing comment? So from here.
Hi, I'm Somiyoshi. As Sensei introduced, I'm co-organizing this MMC seminar. First, thank you very much, Professor Tanaka, for your wonderful talk. So covering from detail to the very comprehensive science. And I also would like to thank also advisors and audience to joining this MMP seminar. Recently, such a mathematical model-based informed drug discovery and development has become to be also mainstream strategy in clinical development and discovery, particularly in the industry. Such a mathematical model can describe, explain the biological systems, pathology, disease progression, drug reaction, and clinical responses. So on the other hand, every model is not perfect. I mean, has some limitation. Therefore, I understand such a model should be treated based on the fit for purpose. So therefore, in this MMP seminar, I hope keeping discussing how such kind of model can be utilized, applied to treat which of the best way. Between the academia and industry, and more interdisciplinary field. Thank you very much.
Thank you very much, Miyo-san. And it is a really good timing. And I really would like my special thanks to Professor Tanaka for fascinating talk and all advisors, very nice questions and online participants. And the next seminar will be held at the end of April next year. And we also invited really, really nice the researcher in this mathematical modeling applying in medicine. So everyone is thank you, and then have a good night, and then good day, and then good morning. Thank you.
Thank you. Goodbye.